Come back from lunch. And this session, I hope, will bring home the idea of how personal treatments that we develop can really be helpful and impactful for patients. So as Jen Wargo has told me before, the battle against cancer is very personal. It's personal for you, for me, and for everyone else whose life has been touched by this disease. We're at a pivotal moment in cancer treatment where research is transforming how we treat and prevent cancer. At the heart of it all are patients who inspire us every day. It is now my privilege to welcome Mr. Joe Easton, who will share how immunotherapy helped save his life. Please come to the stage, Mr. So wonderful to meet you. Thank you.
Well, thank you. Thank you so much for coming.
Well, hello, everyone. Happy Friday. Special thank you to Dr. Alice and Dr. Sharma. Had the privilege of talking with them over a year ago as I started a journey. My name is Joe Easton. I live in Dallas, Texas. I'm married. I'm a father of 4. And my journey started about 14 years ago with melanoma. Originally starting on my ear, and had several reoccurrences over the years, had a couple other places around. And here at MD Anderson, Dr. Jen Wargo was right there with me, helping me through it. Dr. Neil Gross on head and neck. And the good thing at the time is that it had not gone past the surface. But about a year and a half ago, for a routine test, PET scan and CT scan, they thought, well, let's go ahead and make sure everything is still okay. And sure enough, we did it, and sure enough, there was an anomaly that they found in my rib cage, which is very unusual. And sure enough, after biopsy, I had a melanoma tumor in my 4th rib, just over my heart. And it was in my rib, but it was also in the soft tissue around that. And Dr. Wargo said, well, you're going to be fine. She gave me a pep talk, and then she said, I know some people. And again, after talking with Dr. Alice and Dr. Sharma, she connected me with Dr. Tabi. We immediately started immunotherapy. I guess it was December 1st of 2024. 2.5 months, that immunotherapy pretty much got rid of all the cancer cells in the soft tissue and decreased the tumor in my rib by nearly 50% in 2.5 months. So from there, we were excited for the progress, and then I had the good fortune of meeting Dr. Swisher with vascular surgery at MD Anderson. Dr. Swisher removed my 4th rib. This was end of February 2025, and so far so good. It's been an amazing journey. It's now been 8 months. I'm still on a immunotherapy treatment, a reduced amount, but the scans are clear. So we're seeing some good news. So thank you. So I'm not out of the woods yet. We're still on this for a few more months, but I just want to thank you all. This is a wonderful event. Thank you, my family thanks you, and the world thanks you. God bless you. Thank you.
So, Joe, thank you so much for sharing your story. And there's so many patients out there whose stories are very important. And, you know, all of the work that you guys are doing here in this community, I mean, is a broad-based community of scientists and researchers. this is what it's all about. It's about the impact we can have. And maybe it's not your science today, but maybe it's a science that somebody builds on from the work that you did. So I just want everybody to remember, at the end of the day, science is so important, and it's the science that's going to lead to new discoveries. And so we're very happy to have Joe share his story. And for any of the other patients who are in the audience, please know that we're always here to speak with you and to help you in any way that we can. And now we have good news, because it's my pleasure to announce the 2025 poster contest winners. First of all, I want to say to all of you who put your posters up there, what an incredible showing of science here. I mean, it's just been amazing. I don't know if everybody had a chance to go up and see the posters. If you didn't, please, please, please go and see them. I really do think that this is one of the best science that we're doing here at the institution that's at the poster level. So we were able to have judges walk around and view the posters and then make a decision, which I'm so glad I was not one of the judges, because that would have been very hard. But they made a decision to have 3 winners for the poster presentations. And so I'm going to call your name, and you will come up on stage, and Javier has a plaque for you to receive. So
Can I hand them to
the first winner is Mehdi Chaib on immune tetrads predict response to
They did
dual
first or
checkpoint
second?
blockade immunotherapy.
That's first.
And that was for first place. So is he here, or is he still at lunch? Okay. Okay, our second place winner is Falk Ponath from Susan Bullman's lab, understanding the impact of bacterial colonized microniches in the tumor microenvironment.
You're not here either.
Okay, our students must be really still at lunch. Okay, and then our third place winner is Annette Wu from Wen Jiang's lab, leveraging phagolysosome escape for pancreatic cancer immunotherapy. Okay, Jim's going to take home all 3 awards then. Because it doesn't seem like our poster winners are here right now, but we will make sure that they get them. And again, if you didn't get to see the posters, please go on up and get a chance to see them. Thank you. Jim?
What am I doing? Now it's my great pleasure to announce the winner of our very first James P. Allison Institute Catalyst Award, which we want to give to Dr. Giulio Duretta for his visionary leadership that helped make the Allison Institute a real thing. And he continues to work with us to make it even better all the time. Giulio. Where's the photographer? Well, let's get out here.
Come over here.
You should be in the middle.
You got to hold it, Giulio. Thank you. Thank you, Giulio. We love you. Love you.
So I couldn't have done it without.
Okay. Okay, and now we're going to move to the Nobel panel. So they're going to come up on stage. Thank you. Where are you?
Whatever. I jumped the gun. You want to sit? 1, 2, 3. That's it.
Now, Good afternoon, everyone. I'm Alice Park, Senior Health Correspondent at Time, and it is truly my honor to be moderating this panel. It's hard to follow up all the wonderful presentations that we saw all morning, but Pam and Jim, in their infinite wisdom, have figured it out. Invite five Nobel laureates to a discussion. So here we are. I'm really looking forward to it. And I wanted to start with a question for all of you, a pretty basic one. But what drew you to science? What was it about science that attracted you at whatever point, whether you were a young kid or if it came later in life? And maybe we'll start there with Carolyn.
For me, I'd say it would start with my family. Because I grew up in a science household with a dad who was a physics professor at MIT. So that kind of set the tone. And having said that, I don't think I would have admitted to being interested in science until I was in college. And then it was the field of organic chemistry just absolutely captured me.
Okay.
Just like it did all of you, right?
Now, Gary, I know you
Right.
probably don't agree with that love of organic chemistry, but what was it for you?
Well, that's too hard for me. That's hard. You know, the astronomy was something that was pitched to my generation, and so I immersed myself in that, and then when I got to Berkeley as an undergrad, that's when I saw what real science was, but not doing. You know, I never talked to a professor. I don't think in any of the 4 years did I ever talk to a professor, nor do I think that's a bad thing. I mean, you know, like when you go to see a theater production, you don't expect to talk to the actors. These are performers that are teaching you science by performance. My physics professors, math professors, that's what inspired me.
Jim, what was it for you?
I grew up in a little town, and my father was a doctor. You know, did house calls and stuff, and you know, I was a little bit interested in medicine, but also was interested in biology. You know, collecting frogs and horned toads and stuff in the mesquite and cactus fields around Alice, and but there's the Sputnik area, as you were talking about, and I was lucky enough to have the chance to participate in a National Science Foundation program that they had for high school students at the University of Texas. And I went there this summer after my sophomore year in high school and actually got to work in a lab, and I was hooked. After that, I just said, I want to do this. And so when I was 15, I already knew what I wanted to do with my life. So I went and started washing glassware until I convinced a professor to let me work in the lab, and it's all history from there. I ended up going to school and sticking it out, having fun doing science.
So from washing glassware to the Nobel. I like that. And Phil, what attracted you to science?
When I was around 12, I started reading about the history of science and you know, again, some rocketry and the Sputnik era. And it just organized thoughts so well for me. And I thought it was a wonderful way to be able to solve puzzles, learn things, and from then on, that was what I was interested in. Did other things. Math would make that much, was the organizing language of science, and it was just totally almost liberating to be able to work in that dimension.
Okay. And Fred, as the newest Nobel Prize winner, what really drew you to science and the work that you do?
Yeah, I think the first thing was I realized I could not make a living as a professional soccer player. So I was 28. No, I was younger than that. But you know, I was the first in my family to go to college, and I did that because my mother said, "You're really smart. You need to go to college." And so I went to college, and I wasn't sure what I wanted to do. My first year in college, I was a theater major, and I wasn't good enough to be an actor either, so I had to have a fallback. And I always loved science. Science is always great. I even liked organic chemistry. Not enough to actually do it, but I liked organic chemistry. But I took an immunology course in college from Dick Dutton, who I know Jim knows, a lot of people in this room know. And I just got hooked. This light bulb went on, and I went, the immune system's the coolest thing in the world. And it controls all biology and all disease. Not quite true, but more true than not, I think, actually. And I was just hooked. After that, it's like, I gotta work in this field. I gotta understand how this works.
Okay. Now, each of you, really, your journeys to the Nobel started with a what-if question, right? Carolyn, with you, as you said in your wonderful presentation, it was, what if we could see biological processes by connecting them to a chemical reaction and an imaging agent? You know, Gary, with you, it was like, what if there were a different way that mRNA gets expressed? Jim and Fred, I feel like, are kind of connected, because Fred, you were asking, what if there was a way that the immune system recognizes its own cells so that we don't all have autoimmune diseases? And Jim, your question was, what if we could, turn it off for cancer, in the case of cancer? And Phil, it was, what if RNA doesn't get transcribed the way we think, but has these introns and exons? And it seems to get to that point, to ask those critical what-if questions. It's a combination of both critical thinking and creative thinking. So I'd like to also, ask each of you, it seems like there are almost two ends of the spectrum and involve very different skills. How do you see those coming together? How did they come together for you? And we can start at either end here.
Well, for me, the what if was the fact that in the mid-90s, we're starting to do genome sequencing. So we're starting to actually get sequence. And I was never really a geneticist, but I love genetics, because to me, genetics is truth. It'll give you the answer. It may not be useful, you may not understand it, but it's going to be what actually is responsible for something. And so the what if for me was, well, what if we could combine genetics with understanding biology in a way that would be useful for therapeutics. Remember, I was at a company. It wasn't just a pure scientific question for me. It was, how do I get to a therapeutic? And so what we said was, can we use genetics to find the critical nodal pathways that control the immune system? And we found a mouse, and you'll all hear about it later. We found a mouse that let us do that. But I think you're right. It was not just, sequence the genome and we'll have everything, because that sequence the genome was great, but we still don't have everything. And so it was, if we're going to do this, how do we do this in a way that's actually going to get us to something that we want at the end of the day? And that was the twist on the what if for me.
Yeah. And for you, Phil, how do you combine that critical and creative element?
So in my case, it was, what if the nature of a gene in human cells was different than that in bacteria? And there was a hint of very long RNA and short RNA and message. And there really wasn't a question there. There's just an indication that there might be something different. And that led to exploring the pathway by using physical techniques that I had mastered when I was at Caltech. And out of it came split genes, the splicing process. The unusual thing about that discovery was that no one ever phrased the question, are genes split? I mean, that just never occurred to anyone anywhere. And as soon as I solved that, looping of the RNA, I knew there had to be this whole splicing process, and there had to be a whole new layer of biology we did not anticipate and understand.
And that was the creative part, to think beyond what everyone took to be dogma at the time, right?
The creative part was asking that question, because that question had laid in the literature for 20 years, and everybody was ignoring it. And it was, that was the creative part. Once you see it, then you have to tell the story. And you make up the story as well as you can.
Okay. And Jim, how do you bring those two elements together in the work that you've done? Because I think, this whole idea of co-opting the immune system to fight cancer obviously was something that people were like, yeah, makes sense, it should work, but never had before. And how did you bring that critical understanding of how the immune system works with sort of creative ways to say, how do we get it to work?
Well, immunology there was very little known. T cells had been were discovered just a few years before I went to college, and was lucky enough to have an immunology course. And it was mostly about antibodies and B cells, and the last lecture was on T cells, but the professor didn't really, I don't think much was known. He just said they're cells that percolate through your body, go through your blood, your lymphatics, actually go through the tissues and look for stuff that's wrong and take care of it. And I said, how does that work? How do they recognize? He said, nobody knows. No, I don't know. And he finally, I went to talk with him, and I didn't like talking to professors much either, but I went and said, you got to tell me this because I don't understand how this works. And he said, well, he didn't really think there was such a thing as a T cell. He was a B cell guy. He thought it was just a macrophage that had picked up an antibody on its surface, and that was it.
It's hard to believe now, right? That they didn't even think there was such a thing as a T cell.
Yeah, so I just decided I wanted to figure out how they worked. And so I got my PhD, and then I immediately went to an immunology lab for postdoctoral work so I could learn some immunology, and ultimately ended up first just say how they work. First found the antigen receptor, and then the co-stimulatory signal, and then the thing that came along with CTLA-4. Nobody had ever seen a negative signal, an intrinsic negative signal, and turned out that's what that was. A graduate student, Max Crummell, helped figure that out. And the implications for cancer were pretty immediate. This was a pathway that was probably stopping the immune cells from killing tumor cells. And so we said, what if we just took the brakes off? What would happen? And would somebody blow up, or would the tumors go away? And turned out, luckily, the tumors went away. Right. So that was
very important question. Gary, for you, what was that what if question?
I want to stress confusion as a positive attribute. That, when I got to my postdoc, and they were doing nematode genetics, which I was a bacterial geneticist before that, so I knew about bacteria and how to handle them, how to think about them. And they were talking about eukaryotic genetics and crosses and plus over minus and mutant, and had a lingo I didn't understand, and I loved the fact that I was utterly confused. And everybody else knew way more than I did. And it's important because, it seems like we knew what we were doing every step of our careers. And let me tell you, at least I did not. And it sort of accreted over time that I got better at what I was doing. But it took a decade of being surrounded by people who were like way more sophisticated at what I was doing than I was.
But out of confusion comes clarity. So,
or not.
In your case, it did.
Well, sort of. Sort of.
Carolyn, you beautifully explained how you were sitting in that lecture and saw that side and thought to yourself, what if, right? Like, what if we could connect it? Was that a literal eureka moment for you, or was it more kind of this pull and pull between knowing kind of the critical basics of chemistry and biology and then realizing, what if we combine them?
Yeah, I mean, in hindsight, I think what made that story that I already told happen was two things. One is that I had just, a year prior to that moment with Werner Reuter, I had joined this immunology lab coming out of a background in just hardcore organic chemistry. And so I was very naive, and like Gary, I was really confused for the first year in that lab. I didn't know what was going on. But at the same time, there were things that they were doing, and I would say, why can't you just do this? And they would say, well, that's impossible. And I always love when you take someone from outside a field and bring them in and tell them what you think is impossible. And someone from a different field might say, I think I have a way to I think we can solve that problem. So on the one hand, I was the one chemist in this immunology lab watching people struggle with the fact that they had no way to read out cell surface glycans. They just didn't have, and I would say, well, why, isn't there some way we can image these things? I say, no, we can't. And I was like, can't doesn't seem like the right word here, And so my mind was ready to be inspired with a technology idea. And then when I saw Werner Reuter give that talk, that's when it kind of fell into place. So maybe the moral of that story is, first of all, switch fields from time to time and be the outsider, because there's groupthink about what's impossible, and you're outside of that. So you might be able to make the impossible possible, and then be ready to see something and figure out how it might intersect with your problem.
Fresh perspectives, always good. I want to bring in the topic of biotechnology because, of course, Phil, you were involved in the birth of the two first real biotech companies, Genentech and Biogen. And that was when there were two companies. We now have maybe over 20,000, and 200 or so of them are listed on the market. Wanted to get your perspective on, are we at the point now in biotech where perhaps it's a little oversaturated? Are we not being as critical about kind of the ideas that we think are going to be successful? Maybe a loaded question, but it'd be interesting to get your perspective on where the biotechnology industry stands today versus when it began.
So when it began, as beginning means, there was none.
Right.
And we were all into the state that if you asked anyone in the industry of pharmaceuticals or chemistry, you can't do it. All right? Yes, recombinant DNA is really exciting, but 50 years from now, we'll figure it out. And to step forward and try to raise the money and try to do something is a few people, a few places. And then from that time, we've had these up and downs. We're in a very profound down now. There's great technology that you heard about today, discoveries that are going to have big impacts, but the financial markets are uncertain. The FDA and the regulatory framework is different. The healthcare system is changing. It's a moment of uncertainty. And there's been less resources to start something new. I think we're becoming out of it. I think we're beginning to see more money coming in, being at risk for new technology. It's still an opportunity that's in its infancy. It's made great contributions to healthcare. There'll be more, and much more sophisticated. Heard some this morning that were really quite exciting. And there will be contributions to the chemical industry as we rotate from hydrocarbon to more biological processes. There will be contributions in ag that are really happening, and even possibly in some energy sectors. So I think we're still at the stage of trying to understand how we can take this science of genetic manipulation and turn it into organic chemistry. So I was trained as a chemist. I came into this field thinking as a chemist. And when you trained in chemistry in the 60s, half of the people in those labs were going immediately to the chemical industry and take on the position of translating our science into benefit to society. So when I saw the opportunity in biotech, I said, can we make this happen with organic life? And it was just so appealing. I'm totally convinced that we're only beginning to see the impact of all that.
Okay. I want to address another big issue in science today, which is trust. And we've been seeing this degrading of trust in science and scientists. I think it kind of was seeded during the COVID pandemic, and we're just seeing it seem to escalate. So I'd like to ask each of you about, we've seen this before, right? We've seen, the recombinant DNA, when cloning, stem cells, anytime gene therapy, anytime a new scientific breakthrough occurs, there's a lot of public skepticism. Does this period of mistrust in science and the public's sort of almost visceral reaction, right, to the COVID vaccine, to just what science is doing in general, does it seem different from those? And were there lessons from those previous experiences that you think could help us rebuild that trust among the public? I think this is for each one of you, so whoever wants to jump in, maybe we'll start with you, Fred.
Yeah, I don't have the perspective that you certainly talked about in the film about what it was like in the early days of genetic recombinant engineering and stuff. But it does feel a little different to me, and I think that reflects the national discourse on almost everything we have. We don't discuss and debate things. We don't have conversations about things. We have staked out positions, dug our heels in, and on both sides, I personally believe. And so I think it does feel different from what it used to feel. And I agree. I am very worried about not so much the next 3 years. Yes, I'm worried about the next 3 years. Of course I am. But I'm more worried that if this doesn't get reversed, if there isn't some trust and some perceived value, it won't change for a long time. And if people don't trust science and don't think there's value, why would you fund it with taxpayer money? People aren't, right? And I think that's a huge risk, because if there isn't that support, we'll be set back as a nation, we'll be set back as a society, we'll be set back as a planet, generations. So I do worry about it, and I do think it's different right now. What I loved, and this came out in the film, is the conversations you had with non-science people, and when you can get down and have a conversation, sometimes one-on-one or in small groups or whatever, it's better. And I just kind of a parenthetical argument, but the longest interview I've given in the last 2 weeks, and I've given way more than I thought I would ever give in my life, was to the Flathead County Beacon. So you probably don't know where Flathead County Beacon is. Flathead is a county in Montana, which is where I live half the year, and the population in the county is probably 25,000 or something. And I sat down with the reporter for an hour and a half. And she said, I can't believe you're giving me all this time. And I said, you know what? If I can get the word out here in Montana, that's awesome. You're doing it in Bainbridge Island, Washington, I'm talking to like-minded people. Whereas if I'm in Montana, I think it's more important to have a dialogue with people, or at least a subset of which, in some cases a large subset, are on that skeptical side, on that lack of trust side. And so I will spend more time doing that as much as I can. So I do think it's different, and I think we have to take the lesson you guys showed in the early days of genetic engineering and just begin to explain this in a way that gets people to understand the value and the values we have.
And Phil, of course, Fred is referring to those remarkable conversations that scientists were having with local leaders, right, in the Cambridge-Boston area around recombinant DNA when the mayor wanted to just ban it. And of course, Boston was kind of the hub of all of that activity. Are there lessons, do you see, that, from that experience that we can apply today?
I do. We not only talked to leaders, we went to every local neighborhood science fair or festival, set up booths, and talked to people who were there walking the streets. I do think, I agree with everything you've said. The big difference at this moment is that it's the universities that are not being trusted. We have worked 50 years with the working relationships between our research community and basic science and universities, translation to investment in Wall Street, to companies to benefit people. And that really has made a difference in this country's well-being. Rethinking that, we need to gain the confidence of the people in this country at Main Street that this works for them as well as it works for other people. And that's going to take a lot of effort. And we're going to have to be transparent, and we're going to have to execute what we say.
Okay. Jim, any thoughts on that in terms of how we can rebuild this trust among the public?
I think we've got to communicate better. I think in some ways, perhaps the field, we meaning the field, have been victims of our own success. You know, people, they say, why do we need vaccines for? Nobody gets measles. Why do we need vaccines? Nobody gets polio. Well, they've forgotten. You know, I remember when I was a kid, kids with polio, but that's not around anymore, and people forget. And I'll never forget when I saw the TV news of the Surgeon General of Florida saying, nobody can tell me what to stick in my, you know, in the 50s, we didn't ask when people said line up and get the polio vaccine. You know, we knew there was a consequence because we could see it. We knew people had it, and there was no question. You didn't ask whether you were going to do it or not because you knew we all had to do it to protect each other. And that whole spirit is gone somehow, I think, also. And we've got to build that back. Yeah, the greater good. I think the notion that we should do things for the greater good of everyone, that idea itself seems to be receding, if not gone, at least in the minds of a lot of the politicians and people that we see in government these days. You know, it's what have you done for me lately is the only thing, and the long view doesn't seem to really be there anymore. And I think it's frightening because I don't think it has happened before. We've had downturns in funding for science because of a lot of reasons, but not this basic almost animosity and this feeling that we're all, you know, why should I take a vaccine? Why should I worry about this? You know, it's somebody else's problem, you know. I think it's frightening, and I think we have to go out and talk to people again and try to remind them in some way that it is important. And if you stop right now, there are plenty of diseases waiting, not just cancer and more of it, but other infectious diseases that are going to be back, and we're going to be back in a world of trouble, I think. Yeah. So I think it behooves all of us to try to work, but it is kind of scary because the universities used to be a place of respect, and people would listen to what happened, and that seems to be disappearing.
And now they're the target.
Well,
yeah,
they're
the target.
There's now the universities themselves are under attack,
Right.
and I really don't know what the solution is except to knuckle down and try to convince people that, you know, there could be a better world, but we all have to work at it together.
Yeah.
I think.
Gary, your thoughts?
I was recently at the Lasker Awards where Rick Lifton presented the case for cystic fibrosis getting the award this year. And it's an unbelievably moving story. You can look it up at the Lasker site and see the video of him talking about it. And he describes the Cystic Fibrosis Foundation as sort of making sure this happened and sort of mediating support for research and advocating for development of drugs with Vertex as a biotech company. And it essentially cured, it's a major cure in the last 5 years, 8 years, whatever it is. It's a beautiful story, saving the lives of 100,000 people whose lives were miserable in many ways. That's the kind of story that needs to be told, and it needs to be told by those foundations. They raised private money to make that happen. Maybe they advocated for NIH support as well. It's considered extremely effective, but that's a model. And to the other people who complain about wasted money, you know, talk about immunotherapy. Jesus Christ, it's a revolution. It's happened in our lifetimes. You know, shut the fuck up. I mean, what are you talking about? You know.
All right. Carolyn, you know, your work is, I think, a great example of really critical science that now has potential applications that probably were not really obvious even at the beginning, and how important this work is. Now we're looking at ADCs and ACCs. How do you feel about this erosion of trust in science, and your thoughts on just lessons we can learn from our previous experiences on how we can address this?
Well, first of all, I really resonated with what the other panelists have already said. And I do agree, it feels different this time. I'm thinking of the periods of controversy during my own adult life that I can think about and analogize to. But the difference is that, for example, the stem cell controversy of the 1990s, right? There was a time, where we had a federal administration that did not want, that felt it was unethical to do research on human embryos. And research on stem cells was restricted to specific cell lines and things like that. This was before iPSCs, which kind of broke it open in parallel to that. It was certainly motivated by that restriction.
And they even restricted it on embryos that were going to be discarded from IVF.
IVF embryos in
Right.
clinics that had been in the freezer for decades and
Yeah.
were going into the garbage can. But the difference is that I feel like at that time, the concerns were rooted in what I felt were like legitimate, ethical, sometimes religious issues, right? They were, and I say legitimate because the people, who felt compelled to try to restrict research on embryos were people who honestly felt it was bad and were articulating that in an honest way. And same with fear around recombinant DNA technology, right? It was an unknown, it was a new thing, it seemed frighteningly powerful. And you can fast forward to the CRISPR ethics, debates that have been around, which, should we be doing CRISPR engineering in human embryos and in human cells? And I think that there's a legitimate set of concerns and a legitimate debate. And even if some of the concerns are not rooted in science, they might be rooted in culture, they might be rooted in religion. That's still legitimate. People are speaking from the heart and they want to talk it out, They want it to slow down so that they can have time to think about it and talk it out, right? But now that is not the feeling that I'm getting. I feel that the anti-science rhetoric coming out of Washington is very cynical. I don't think that some of the loudest voices that are arguing against supporting biomedical science actually, I don't think that they feel in their hearts that science is bad, that universities are bad. They send their own children to universities. Cystic fibrosis is a really good case in point, because one of the loudest voices in our administration who's working the hardest to undercut university activities is a person whose child is being treated with the Vertex drugs for cystic fibrosis, right? Who benefited directly from decades of NIH-funded science and biotechnology sector.
And how many of them probably have had loved ones treated for cancer?
Right, and they know this, okay? So the forces that are undercutting science right now are doing so, to my mind, okay, they're doing so not because they have legitimate ethical concerns, religious concerns, cultural concerns, or just fear of something new. It really is very cynical, and it's a mechanism to acquire power. So that's the problem, because you can slow down things, you can have discussions and debates, but nobody's mind will change if their opinions and their actions are not based in truth. That's the problem.
Good faith. Yeah.
Okay. I can keep asking questions, but I know we're already over time, but I do want to open it up very quickly to perhaps one question from the audience. I know it's a very unique opportunity to have these Nobel laureates here, so if there's a question from the audience, we'll take a quick one. Yes.
So thank you, panelists. My question is for the entire panel. In today's research environment, there's a strong focus on translational science and quick application. I'm just wondering, how do we ensure that we continue to protect and fund purely curiosity-driven basic research, even when the practical value isn't immediately obvious?
Thank you for that question. Yeah. Should we start with you, Fred?
Yeah, it's a great point. You will hear in a little while that the reason I got the prize started in the Manhattan Project. And so when I talk about the fact that it takes 50 years sometimes for basic science to become useful. And I put that in quotes because you can define that any way you want. But how do we ensure that? I think is a really difficult scenario, because I think you're right. People look to short-term gains right now a lot. And historically, the federal government has been the group that has supported, the long game. And so I think we just have to come back, and Carolyn was talking about this, we just have to come back and try to ensure that at that level, it may not be at the biotech level. I don't think they're ever going to take the 20-year, well, I shouldn't say ever, but most biotechs aren't going to take a 20-year time frame anymore. But the federal government can and should, and I think we need to make the case that they continue to do it. It's got to be the federal government, and some private foundations will do it as well, and I think that's okay. But we've got to target the right audience for that.
Can I add something to that?
Yeah.
Because whenever I give the talk that I gave, and I talk about 100-year-old chemistries that were the basis of what we did, I have to accept the reality, which is that the chemists, 100 years ago, that did the work that was the backbone of what we did, like there was no way that they would have anticipated, 100 years from now, somebody's going to image glycans with my chemistry. Right? They did the work because these were molecules that were weird, and they were curious, and they played around with them and published, And so I think if we're all being honest here, we're sitting here because a panel of Swedish scientists decided that the work that we did sometimes 20, 30 years ago was impactful. But most of us will not know the impact of our own research in our lifetime. That's just the truth. So there's a leap of faith that you want to articulate, and how you convince the government to support a leap of faith. That's a hard message, but it's the truth, right?
It's not a hard message. As you say, it's 100 years of evidence that basic research is absolutely essential to making progress. We have large gaps of unknown. We have to develop that unknown and discover what's there. And inevitable, it will lead to a more truthful, honest, and economic, better life. And we just got to keep saying that it will be there. We have to have basic research, particularly in areas such as life science, that are really new and unexplored by and large. So it's essential.
Okay. Gary, any thoughts to add?
NIH doesn't do a very good job of selling science to the American public. I spent a lot of time with NASA, and I got to see how masterful they are at selling science. Now, space is easier to sell than a mitochondrion.
Or a worm, for that matter.
But they do it, and that's a model. And NASA needs to learn how to sell it to the public and say, what does it cost? What's the benefit? And tell people. And the Nobel Prize can be useful because you can say, well, why is it that a quarter of the people, or whatever the number is, a third, are Americans, or that says something about American investment. It's not that we're genetically superior. We spent more dough.
Right. Jim, I want to give you the last word here on...
As Phil said, there's a lot we don't know, and we're not going to know it except people that curiosity diving in to try to shine light on the things we don't know, and that's the ultimate.
Yeah.
That's the ultimate science there. It's just purely curiosity-driven science, and I think there's a lot still be done, and we can't lose track of that.
Yeah. Back to that what-if question.
Yeah.
Well, thank you, all of you. I really appreciate it, and I hope we can do this every year. This is fascinating. Thank you.